welcome to Biology, the scientific study of life. The main stages of food processing are ingestion, digestion, absorption, and elimination. We begin our study of nutrition by serving the diverse feeding mechanisms that have evolved among animals. But ingestion, the act of eating, is only the first stage of food processing. Organic material on food consists largely of proteins, fats, and carbohydrates in the form of starch and other polysaccharides. Animals cannot use these molecules directly for two reasons. First, polymers are too large to pass through membranes and enter the cells of the animal. Second, the macromolecules that make up an animal are not identical to those of its food in building their macromolecules. However, all organisms use common monomers. For example, soybeans, fruit, flies, and humans all assemble their proteins from the same 20 amino acids. Digestion. The second stage of food processing is the process of breaking food down into molecules small enough for the body to absorb, as shown in this figure. Digestion cleaves macromolecules into their components monomers, which the animal then uses to make its own molecules or as fuel for ATP production. Polysaccharides and disaccharides are split into simple sugars. Fats are digested to glycerol and fatty acids. Proteins are split into amino acids, and nucleic acids are cleaved into nucleotides. Recall from chapter 5 that a cell makes a macromolecule by linking together monomers. It does so by removing a molecule of water for each new covalent bond formed. Digestion reverses this process by breaking bonds with the addition of water. The splitting process is called enzymatic hydrolysis. A variety of hydrolytic enzymes catalyze the digestion of each of the classes of macromolecules found in food. This chemical digestion is usually preceded by mechanical fragmentation of the food by chewing. For instance, breaking food into smaller pieces increases the surface area exposed to digest juices containing hydrolytic enzymes. The last two stages of food processing occur after the food is digested. In the third stage, absorption, the animal cells absorb small molecules such as amino acids and simple sugars from the digestive compartment. And finally, elimination occurs as undigested material passes out of the digestive compartment, as shown in this video. Notice the four stages of food processing. First one, ingestion, the food. Digestion, mechanical digestion, pieces of food. Chemical digestion, enzymatic hydrolysis, small molecules. This is absorption. And nutrient molecules entry, food cells. The last is undigested material is elimination. Digestion or digestive compartments. How do animals apply their digestive process to food without digesting their own cells and tissues? After all digestive enzymes hydrolyzes the same biological materials such as proteins, lipids, and carbohydrates that animals are made of, and it is obviously important to avoid digesting on self. Most animals reduce the risk of self-digestion by processing food in specialized compartments. Intracellular digestion, food vacuoles, cellular organelles in which hydrolytic enzymes break down food without digesting the cell's own cytoplasm, are the simplest digestive compartments. This digestion within a cell, called intracellular digestion, begins after a cell engulfs food by phagocytosis or pinocytosis. Neatly formed food vacuoles fuse with lysosomes which are organelles containing hydrolytic enzymes. This mixes the food with the enzymes, allowing digestion to occur safely within a compartment that is enclosed by a protective membrane. Sponges are unusual among animals in that they digest their food entirely by the intracellular mechanisms. Extracellular digestion. In most animals, at least some hydrolysis occurs by extracellular digestion, the breakdown of food outside cells. Extracellular digestion occurs within compartments that are continuous with the outside of the animal's body. Having an extracellular cavity for digestion enables an animal to devour much larger prey than can be ingested by phagocytosis and digested intracellularly. Many animals with relatively simple body plans have a digestive sac with a single opening. This pouch, called gastrovascular cavity, functions in both digestion and distribution of nutrients throughout the body, hence the vascular part of the term. The cnidarians, called hydras, provide a good example of how a gastrovascular cavity works. Hydras are carnivores that capture prey with specialized organs called cnidaria, and then use tentacles to stuff the food through their mouth into their gastrovascular cavity, as shown in this figure. Specialized gland cells of the gastroderms, the tissue layer that lines the cavity, then secrete digestive enzymes that break the soft of tissues of the prey into thin pieces. Notice that. Other gastrodermal cells, called nutritive muscular cells, engulf these food particles, and most of the actual hydrolysis of macromolecules occurs intracellularly, as in sponges. After a hydra has digested its meal, undigested materials remaining in the gastrovascular cavity, such as exoskeletons of small crustaceans, are eliminated through the single opening, with functions in the doyle roll of mouth and anus. Many flatworms, also having, as shown in this figure, have a gastrovascular cavity with a single opening. In contrast to cnidarians and flatworms, most animals, including nematodes, annelids, mollusks, arthropods, echinoderms, and chordates, have a digestive tube extending between two openings, a mouth and an anus. Such a tube is called a complete digestive tract or an alimentary canal, because food moves along with canal in a single direction. The tube can be organized into specialized regions that carry out digestion and nutrient absorption in a stepwise fashion, as shown in this figure. Notice esophagus, stomach, and GZ, intestine, and anus, crop, mouth. Stepwise fashion. Another advantage of a complete digestive tract is the ability to ingest additional food before earlier meals are completely digested, which may be difficult or inefficient for animals with gastrovascular cavity.